Hello friends, welcome to Sam Biology class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about glycolysis. The term glycolysis is derived from two Greek words. Glykos means sugar, lysis means loosening. It is alternately known as Emp Pathway to commemorate the works of Gustav Embden, Otto Meyerhof and Jacob Parnas. Glycolysis, one among the most completely understood biochemical pathway, is a sequence of 10 enzymatic reactions in which one molecule of glucose is converted to two molecules of 3-carbon compound known as pyruvate with the concomitant release of 2 ATP. Glycolysis can be divided into two stages, stage 1 and stage 2. In stage 1, there is energy investment. In this stage, one molecule of glucose is phosphorylated and cleaved to produce two molecules of glyceraldehyde 3-phosphate. This process consumes 2 ATP. In stage 2, there is energy recovery. In this stage, two molecules of glyceraldehyde 3-phosphate are converted to two molecules of 3-carbon compound known as pyruvate with the concomitant generation of 4 ATP. Glycolysis therefore has a net profit of 2 ATP per glucose. Stage 1 consumes 2 ATP, whereas in stage 2, 4 ATP are produced. Reaction steps of glycolysis. In step 1, alpha-D glucose is activated or primed by phosphorylation at carbon 6 to yield alpha-D glucose 6-phosphate using ATP as the phosphate group donor. This reaction is catalyzed by the enzyme hexokinase, which is present in plants, animals and microorganisms. Another enzyme exclusively present in liver, known as glucokinase, also catalyzes this reaction. But glucokinase is specific for glucose. It do not act on other hexoses. Glucose 6-phosphate is an important compound being present at the junction of many metabolic pathways such as glycolysis, glycogenolysis, gluconeogenesis and the hexose monophosphate shunt. Step 2 of glycolysis. Step 2 is the isomerization of alpha-D glucose 6-phosphate to alpha-D fructose 6-phosphate in the presence of enzyme phosphoglucose isomerase. This reaction is an isomerization reaction in which the 6-membered pyranose ring of alpha-D glucose 6-phosphate is converted to 5-membered furanose ring of alpha-D fructose 6-phosphate. This isomerization reaction involves a shift in the position of carbonyl oxygen from carbon 1 to carbon 2. In step 3, alpha-D fructose 6-phosphate is phosphorylated to form alpha-D fructose 1,6-bisphosphate using ATP as the phosphate group donor. This reaction is essentially irreversible and is catalyzed by another inducible enzyme, that is phosphofructokinase, which catalyzes the transfer of phosphate group from ATP to the carbon 1 of alpha-D fructose 6-phosphate, thereby producing alpha-D fructose 1,6-bisphosphate. Reaction 4 is an aldol cleavage. In this step, alpha-D fructose 1,6-bisphosphate is cleaved between carbon 3 and 4 to yield one molecule of glyceraldehyde 3-phosphate and another molecule of dihydroxyacetone phosphate. Both are triose phosphate, but Glyceraldehyde 3-phosphate is an aldose, whereas dihydroxyacetone phosphate is a ketotriose. This cleavage is catalyzed by the enzyme fructose bisphosphate aldolase. Step 5. Only one of the products of the aldol cleavage reaction, that is glyceraldehyde 3-phosphate, can be degraded directly by the subsequent reactions of glycolysis, whereas the other product, that is dihydroxyacetone phosphate, cannot. However, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are interconvertible by isomerization reaction, which is catalyzed by the enzyme triose phosphate isomerase. This is the last reaction of the stage 1. So we can say that from one molecule of glucose, two molecules of glyceraldehyde 3-phosphate are produced in the stage 1. Stage 2 of glycolysis. The first reaction of stage 2 is the oxidation and phosphorylation of glyceraldehyde 3-phosphate by NAD plus and inorganic phosphate in a reaction catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this reaction, aldehyde oxidation, which is an exergonic reaction, drive the synthesis of high energy acyl phosphate, that is 1,3-bisphosphoglycerate. Acyl phosphates are compound having high phosphoryl group transfer potential. Reaction 7 yield ATP together with 3-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase, which catalyzes the transfer of a high energy phosphate group from the carboxyl group of 1,3-bisphosphoglycerate to ADP, thereby resulting in the formation of ATP. This is the first reaction of glycolysis that generate ATP. In next reaction step, 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. Mutase is an enzyme that catalyzes the transfer of a functional group from one position of a molecule to another position. In reaction 9, 2-phosphoglycerate is dehydrated in the presence of enzyme enolase to yield phosphoenol pyruvate. Reaction 10 of glycolysis. Reaction 10, which is the last reaction of glycolysis, is the conversion of phosphoenol pyruvate to pyruvate. This reaction is catalyzed by the enzyme pyruvate kinase, which catalyzes the transfer of the high energy phosphate group from phosphoenol pyruvate to ADP, thereby resulting in the formation of ATP. This is the second reaction of glycolysis which generates ATP. Glycolysis is a metabolic pathway by which each molecule of glucose is partially oxidized, yielding two molecules of pyruvate. This is the overall reaction of glycolysis. The products of glycolysis are ATP, NADH and pyruvate. In stage 1 of glycolysis, 2 ATP are consumed, whereas in stage 2, 4 ATP are produced by the process of substrate level phosphorylation. Glycolysis therefore has a net profit of 2 ATP per glucose. In some tissues and organisms for which glucose is the primary metabolic fuel, the ATP generated by the process of glycolysis satisfy most of the cell's energy needs. In glycolysis, for each molecule of glucose, 2 NAD plus are reduced to to NADH. Reduced coenzymes such as NADH represent a source of free energy which can be recovered by their subsequent oxidation. Electrons from reduced coenzyme passes through a series of electron carriers to the final oxidizing agent molecular oxygen in a process known as electron transport. The free energy of electron transport drives the synthesis of ATP from ADP and inorganic phosphate by a process known as oxidative phosphorylation. This event also sought to regenerate NAD+, which can participate in the further rounds of reactions catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Under anaerobic condition, the NADH must be oxidized by some other means to keep the glycolytic pathway supplied with NAD+. In glycolysis, glucose is partially oxidized to pyruvate, and pyruvate is a still relatively reduced molecule which undergo complete oxidation by the citric acid cycle. The free energy released during this process drives the synthesis of much more ATP, whereas in anaerobic metabolism, pyruvate is metabolized to a lesser extent that help to regenerate NAD+. So friends, this is about glycolysis. For more updates, please subscribe Science Biology class and also please share your comments. Thank you.